All right, welcome to TIL Talks with Chris White, where we talk TILs, tumor infiltrating lymphocytes, and all advanced cell and gene therapies, both with patients and caregivers, along with industry leaders. Let's get going. As a reminder, I am not a medical professional or medical expert. I am only a professional and expert on my story and my experience. If you are needing or seeking medical advice or assistance, please reach out to your care team. All right, we are now rolling. What's up, everybody? Welcome to TIL Talks. I ecstatic to introduce my guest today, the first of the type in the series that I've been doing for TIL Talks. This is one of the industry leaders that I mentioned from time to time. This is Mr. John Powderly. We met in March, I want to say, at the Deloitte Next Gen meeting in Westlake, Texas, and got to know each other a little bit. And I'm super ecstatic to have you on today because you were talking to me about something that is TIL, but that I hadn't heard before, and it just got me really intrigued. And if I'm correct, it's called Peripheral TIL. But I would like to have you give me a one-on-one on who you are and what you do. Let's kind of just go from there.
Yeah, thank you, Chris. It's good to see you again, and hope to see you at more of the Deloitte Industry Working Group meetings. The rest of the industry is talking about how to do personalized cell therapies. So I'm an oncologist. I'm kind of a rare bird because I have a dream job. I get to see cancer patients and be motivated to do research trials and help to develop immunotherapy drugs. So offer clinical trials at our clinic. We're a phase 1 clinic. We're called Carolina Bio-Oncology Institute. We're here in the Charlotte suburbs of Charlotte, North Carolina, and I've been doing phase 1 clinical trials now for about 20 years. Our claim to fame is we helped to develop the early checkpoints. So the drugs that were on TV, Super Bowl commercials, we helped to give the first dose of many of those and have worked with, I would say, most of the drug companies developing checkpoints. And over the past 20 years, we've done about probably 180 phase 1 studies and seen over 5,000 cancer patients. So we have a mission to help accelerate access to early phase immunotherapy studies, not only drugs like antibodies and checkpoints and vaccines, but also developing cell therapies, which is how we got on our discussion. And you're a phenomenal TIL survivor. And we chatted about melanoma patients who survive with TIL or immunotherapy checkpoints. They get the vitiligo and the pigment change. So that's the highest good prognosis of a durable response. So that's great. What you got, the TIL therapy at your time, the surgically resected TIL, tumor infiltrating lymphocytes, what you got was the types of TIL from a surgical resection that my mentor, Steve Rosenberg, helped to develop about 35 years ago. And that's where they can take the tumor out, surgery, remove it, and they grow up the immune cells with cytokines like interleukin and other cytokines. The cells proliferate in the 100,000 plus fold, and they'll give those back to you as a dose. And those TIL cell therapies have been proven to work not only for melanoma, but a couple other tumor types. So it's very exciting. That was the first generation of TIL cell therapy. It's super promising, and you're a living proof of the durable responses. So the programs we're working on here are cell therapy in TIL analogs or TIL-like, but they're not surgically resected from a tumor. Instead, the technology now has become so forward that we can now detect in the bloodstream of patients early TIL or peripheral TIL. We say the peripheral because they're TIL floating in the bloodstream. So they are the immune cells that are in the tumor, but they've leaked out of the tumor and they're floating around in the bloodstream. And there's many different ways they've been described in the literature. Peripheral TIL is the easy way to explain to somebody who knows what TIL is. Say, oh, it's the peripheral TIL. It's the TIL, not just from one tumor, one resection, but it's those immune cells floating around that are cancer specific. The other terms you'll hear about these peripheral TIL is that they're cancer experienced or cancer educated, or they're exhausted. You'll hear them be tumor reactive. So there's many terms used in the literature about what are they. And we know that they're neoantigen specific. Neoantigens are the mutations that are within the tumor. So these peripheral TIL are with any solid tumor, just like there's often TIL in the tumor, the peripheral TIL float in the bloodstream, and they might only be like 0.1% to just about 1% of the bloodstream. So if you can obtain those immune cells, those lymphocytes, those peripheral TIL, and you can identify what are their markers and how can you select them or grab them from the blood or from leukapheresis, which is where you can donate, like the Red Cross, you can donate blood and identify them and then grow them up and test them against the patient's own tumor. You can verify that indeed you've got the peripheral TIL, and you can expand them and give them back as a dose to patients, or you can bank them for future use. And so that is it's kind of a new field that is a derivative of the original TIL industry. And I'm estimating there's probably about 4 or 5 companies now working on that. We're one of those companies that are developing that technology. And what is special about our model is we are a phase 1 clinic. That's my day job, is seeing patients doing immunotherapy drug trials with biopharma industry. But during the evenings or after hours, I employ another team with another sister company where their full-time day job is growing peripheral TIL, isolating them, testing them against patients' tumors so we can verify the peripheral TIL, studying the markers, trying to making them more reinvigorated, and then looking at other immune cells besides peripheral TIL that may also kill cancer. And we call each of those different types of immune cells fractions. They're different fractions of a bag of blood or a leukapheresis product. And the other way we look at them is Army, Navy, Air Force, Marines, right? They all kill their target, but they have different mechanisms, different ways they may hunt or find the cancer, different waves of attack. And so this is a new field, and it's exciting. Your audience may have heard of some of these other fractions of immune cells that are kind of in the news if they're looking at trials. One other fraction we're working on, which is kind of our next after the peripheral TIL, our next fraction is natural killer cells, which we're seeing those more and more in Asia. And now there's probably about 24 companies in America working on natural killer cells. And there's a version of natural killer cells we're working on called memory natural killer cells. And we published a paper on that about 2 years ago. They kill a little bit differently and they kill different types of tumors, but they're in the fraction that is valuable to patients who want cell therapy. The other fractions that some of your listeners may have heard are, instead of TIL are usually alpha beta T lymphocytes, which is the most common T lymphocytes. It's a sub fraction of alpha beta T lymphocytes are typically the TIL. But there's other peripheral TIL or peripheral immune cells called gamma delta T cells. And gamma delta T cells are very interesting because they are evolutionarily much older than the typical human alpha beta T cell. So most of the T cells you got in your TIL product are alpha beta T cells. And those are evolutionary newer cells that have been around in biology since many mammals. And we know that they are indeed important to kill cancer. They're often the cause of the vitiligo, and they target specifically tumor antigens or neoantigens. When alpha beta TIL, like what you had, when they target tumors, they target those tumors in the context of self, meaning they depend upon an MHC molecule to present a specific antigen, which is a good way to fight viruses or to fight cancer if you have a good immune system. Compared to the gamma delta T cells, which is the fraction, those are special because they don't need to have tumor antigens presented to them in a way that is the context of self. So those gamma delta T cells can recognize a much broader repertoire of tumor antigens. So those are promising. So we have other tumor fractions we're working on. We can continue to talk about that in a little bit, but that's the type of work we do. And our goal here is to be able to do all these fractions from a single patient for autologous cell therapies without needing to go to big surgeries, but to be able to do it in a research clinic with the different fractions, kind of like a sequential dosing scheme where we can be the Army, Navy, Air Force, Marines of these different fractions coming in and working at different angles to try to kill the cancer, all autologous and all chosen fractions that we know do the best killing based on each patient's tumor types.
Super exciting stuff. I mean, man, I got a list of like questions, and I know I've kind of brought some of these up with you before, but in a sense, what you're doing is, the way I kind of see it, is a ad hoc or like a hybrid model of what would be considered a decentralized version of a centralized, larger scale form of like TIL, but it's one, it's personalized, but two, this is something that you were describing to me before as like kind of similar to like a vaccine or an mRNA type of product, to where you would come in every so often, and instead of doing a typical or traditional IV infusion, you get it through a shot. Correct?
So yeah, the cell therapies like the peripheral TIL or your TIL are given through an IV. Some of the other individualized or personalized vaccines, like there's mRNA vaccines as an example, or other individualized antisense, which is a way to like silence genes, silence gene drivers. Those can be given by a shot or by an infusion. Those antisense oligonucleotides, those are also in development. They can be very personalized for patients. And just like cancer vaccines now are becoming more and more personalized based on each patient's individual tumor mutations. And there's the good news and the bad news. The good news is, yes, the technology is moving in that direction, but they're highly logistically complex because you have to manufacture batches for each patient's individual tumor mutations.
One at a time, right?
One at a time, and they're small batches because they're for individual patients. So there's a lot of logistics in that. In the current models, as you hinted at, they're centralized or decentralized. The current biopharma model is mostly what we're seeing in the globe is mostly like how you got your TIL. They got your surgery was resected at one hospital. It was sent on ice to another facility. They manufacture it at that one facility, maybe East Coast and West Coast, 2 facilities. And then they ship it back frozen to the clinical site to thaw and dose the patient. So that works. That's the first generation of TIL, like what you got. What we're seeing starting to happen in the field is, and we're kind of leading the efforts on this, is more facilities, and some of them are the hospital systems that are trying to do it decentralized, like a Moffitt or an MD Anderson or those types of, they're trying to grow their own TIL, or doing them in our case as an outpatient setting. That's what we're working towards, is outpatient peripheral TIL to make it easier and more affordable, but where the logistics are all contained at a regional center that we plan to blueprint these regional centers around the globe. So, and the same goes for the other individualized medicines that are now in development, like mRNA vaccines or antisense oligonucleotides. Eventually, I think the best way to deliver these to the masses of patients as the technology evolves and as the industry matures is more decentralization, so that these can be more than just 1 or 2 facilities in each continent, but rather a medium-sized facility in each city, and can deliver these types of promising individualized cell therapies or individualized peptides or oligonucleotides or mRNA vaccines. So that's where we see the future is going. There's a lot of challenges. We can get into the challenges old, but that's where we see the future going.
Like in simplified form, I always just think of it like, I call it stupid simple or simple stupid, where it's like if Nordstrom as a giant facility has a centralized mall location where you can get like everything that they have all in one place, but then they also have what's called like Nordstrom Rack, where it's like those little outlet malls that they have like a handful of their products, at these other spots, so then they can put them in places that you wouldn't normally see them, and it brings the availability for people to acquire those. And so that's kind of how I would like to see, or what I am seeing with cell therapies, because the traditional models, or a lot of them, are some centralized locations. But if from the patient perspective, you could take like one portion out of that entire cycle of everything that needs to happen in order to make it happen, that's one less step. If there's any part of it to where it needs to be frozen and then thawed and then frozen and thawed again, if you can reduce one of those freezing and thaws, like, the better. Imagine a grape. This actually came up at Deloitte when I was having a conversation with the gentleman, and he was just like, think of it like this. Say you take a raspberry or a grape, and you pull it off the vine, and then you freeze it and ship it and then thaw it, and then like a cell or a tumor, and then you do what you got to do to it, and then you have to freeze it again to ship it and then thaw it. When it's finally thawed out for the second time, it's probably gonna have the shriveledness and some of the after effects from doing multiple freezings. And from a patient, like if you're not within X amount of miles or whatever of a location, then you got to travel to it. And so that's an extra step. That's some more logistics. That's more planning. But I always kind of thought of, well, what if there was a certified surgeon? This is in the case of like solid tumor TIL, like I did, obviously, to where in my case, I had to go from Dallas to Denver to have my tumor resected, and then it had to be shipped to the East Coast to be expanded and then shipped back to Denver. And then in the meanwhile, I go back to Dallas. But let's just say, what if there was a certified surgeon that had like the correct qualifications and nurse or anesthesiologist? It was a part of his traveling team that could come to my local place, whether I was in the DFW Metroplex or in some rural city that had like a care clinic that could come to me, do my correct resection, and the companies that contracted to ship, they can show up from almost anywhere and then ship it. But that's one less step for a patient to have to go to the location and then to ship it. And so it just kind of reduces the logistics. And on top of all that, it reduces the time.
Yeah.
And that's, I am so big on that, as time is the biggest currency. Like if you can take a 22-day process and knock it down to 14 days or even to 7. And I remember specifically as we were having breakfast one morning, you were kind of describing the brick and mortar locations, but being able to do it in kind of these smaller locations and more the kind of rural spots where people could access them more easily that they wouldn't normally be able to do unless they traveled multiple miles. And so it was kind of, it wasn't, full out centralized or full out decentralized, but it was kind of like, a compromise between the two.
Yeah. No, you're touching on a lot of good points. Like specific to the freeze thaw, it's known amongst the TIL immunotherapy docs and anybody that works with lymphocytes or immune cells. Anytime you go through a freeze thaw, you lose about 20% viability. So if you're doing a freeze thaw at each shipping, receiving, back and forth from the factory back to the clinical site and leukapheresis site, you're losing 20 plus 20% viability. And you potentially, we have data that you lose even more when it comes to functionality or specificity. So maybe that you're killing kind of the more valuable cells. And sometimes the most valuable cells may be the more exhausted ones that have been fighting the cancer more specifically, the more experienced ones. I mean, they're the Marines that have been in the battle the longest. They may be more valuable and more sensitive to freeze thaw. So our model, the decentralized model that we're really in the point of care camp. Like we're so decentralized, it's the point of care. I mean, there is patients coming to the same facility, the clinic's in the front, the manufacturing facility's behind me. So it's the same facility all under the same roof. In that model, there's no freeze thaw. It is warm change. Here in North Carolina, we're all about speed. NASCAR, how fast can you change a tire?
Right.
And then 25 seconds and you're. So for us, when a patient comes in and they leukapherese early in the morning, we can walk down that leukapheresis product down the hallway in a warm chain. While it's still warm, we can basically put it in media, look at the qualify, characterize it, give it the growth media and give it the stimulation it needs, all the reagents. And so we can do those things back to back. And then the same comes when we check later and it's time for harvesting. We can also walk the cells back down the hallway as we get them ready for dosing. So that proximity makes it easier logistically, saves money, saves costs, less freeze thaw. The patients have a little more flexibility on what days they can pherese, what days they can get dosed. It also allows something really valuable, and that is if we have the proximity to the patient, we can also get a sample of their tumor and grow their tumor in parallel, and then test to confirm that we've got the right population of peripheral TIL or gamma delta or NK cells doing the best killing. That lets us pick the best fraction or the best subfraction. That's what's phenomenal. So we could tell a patient on, earlier in the process, hey, on a small scale, this worked best. Let's upscale this process. And also being at the point of care for doing cell therapy like this, it lets us iterate it better. For example, a lot of the current cell therapies that are FDA approved on the market, it's a one-time dose, one and done. And then you just pray it works. And luckily, it does work. But in a point of care model like ours, where we have access to the tumor, access to the patient, access to another batch of cells, if that tumor partially responds and then mutates away, we can repeat the biopsy, repeat the leukapheresis, grow a new batch of immune cells, and try to recapture the mutant clone that mutated away. They call that tumor escape. So that's a big part of the resistance that happens with immunotherapy. So with our model, point of care model, and specifically having the patient is kind of the custodian of their cells down the hallway we're growing for them as a professional service. It lets us keep costs low so we can iterate it more quickly. If it doesn't work the first time, we'll make it better the next time or try a different fraction or a different stimulation method. So it's kind of like a perpetual trial that's ongoing. And we think that resonates more with patients who want access to the cutting edge, the prosumers who want access to cutting edge research on early phase trials.
Gotcha. I wanted to ask if, because I've heard it both ways, but in regards to being able to extract and expand cells, I've had solid tumor cancer patients do both just a tumor extraction, and I've also had them tell me they've had to do a tumor extraction and a nephoresis as well. And then I've had a lot of hematologic cancer patients say that the only way is, just they're doing a nephoresis, double arm in the chair. But the way that your model is described to where, there is no having to freeze it and ship it, is there, any kind of, specific consensus on what would be more preferred? Or is there like, you have to do it if it's this type of disease or cancer, or any kind of hybrid model, or is that being studied in what you're doing?
Great question. If you look at the hematologic cancers, like the leukemias, lymphomas, technically that cell therapy product that they get is not a TIL. It's not a tumor-free lymphocyte. It is really is. It's one of their bulk immune cells, a group of their bulk immune cells that could have been otherwise targeting anything, some other old memory of some old, virus or something. And what they do is they put in a gene construct into that.
They had a protein and edit it, right?
Yeah,
Or
the CAR T. So they put in a gene construct so that it's basically forcing that immune cell, that bulk immune cell with the gene construct now, to give it a surface receptor that will go and target leukemia and lymphoma specifically. That's the so-called CAR T, the chimeric antigen receptor T cells. So that it's great technology as the first generation. It works. Response rates like 80-90%. It works, but it's a one-time dose. It's a lot of toxicity can come with it. It really broke the new wave into the cell therapies, especially for hematology, leukemias, lymphomas.
yeah.
Wonderful treatment. Those are still all manufactured. Well, not all, but most are manufactured centrally. They're shipped and they're frozen and thaw. Freeze thawing might not matter as much for those because the gene construct has a gas pedal in it, and the cells are going to
Right.
proliferate in the patient. So when it comes to TIL, for solid tumors, the TIL are polyclonal by nature, not a single gene construct targeting one thing like the CAR T for leukemia and lymphoma, targeting a public antigen like the CD19, which is the target, one of the many targets for leukemia lymphomas. On the solid tumor TIL or peripheral TIL or NK cells, they're polyclonal, and they really are a slurry of many different clones targeting many different epitopes or antigens or surface of the patient's tumor. And those cells may be a little more sensitive to the freeze thaw. They typically don't have a gas pedal in them, a gene construct. They're more natural, if you sense. They've been reinvigorated, they've been grown up. They're a little more sensitive to freeze thaw. They're a little more challenging because it does take a lot of reinvigoration and a lot of balancing of their environment to make them grow healthy and robust and to give them back. And then when they give them back, as you know, they're chased with some immune growth factor stimulators like the cytokines, like what you got. And they're given after some doses of chemo to knock down the resident immune cells to make room for the new donor TIL. So that's the current, I would say, first generation TIL that works good. When you look at where the paradigm may be shifting and where the industry may be going is the infrastructure is not built out yet for decentralized cell therapy countrywide in the United States, much less point of care. I think we're the only real true point of care model trying to get rolling on piggybacking on a phase 1 clinic down the hallway. But when you look overseas at some of the facilities in Asia and in Europe, they really are doing point of care manufacturing or across the street manufacturing in outpatient settings, but associated with regional hospital facilities. And their regulatory agencies have been approving those differently than what the FDA regulations are here in the United States, because a lot of those are defined cell therapies based on if they can be grown in so many days, if there's not a gene construct, if it's autologous, and if the hospital has been trained and certified, it's looked at a little bit more like a professional service. So in that aspect, some parts of Asia and Europe are a little more advanced and forward thinking with their infrastructure on building
Oh
out
yeah.
distributed models, decentralized models, point of care. I think America will catch up a little bit. Some of the larger academic centers and larger hospital systems in America are already looking at how can they get the tabletop devices to grow CAR T, work with partners to teach them how to do it, how to license the products or the gene constructs. And we're doing that not for the CAR T, but we're doing it for the solid tumors for those different fractions. I think we're the only company looking at all those different fractions, trying to grow them in parallel to choose the best ones. But yeah, the industry still has a long way to go. The other challenge is that if you look at, this is part of, these are tough lessons I've had to learn. Individualized or personalized medicine sounds great on papers. It's fantastically futuristic. But when it comes to like trying to get investors to fund it,
Yeah.
most biotech venture funds, firms, they want to get a return on their investment in like no more than 5 years. Just to develop a cell product, much as any drug, is really a 5 to 10 year
Let
product.
alone make it a commercially,
Let alone
available thing
sustainable,
to
commercial, let
get
alone
an
build
ROI.
let alone build the infrastructure. Like you have to build a building for each facility. That's dirt, that's bricks and mortar, that's, multiple different PhDs on a regulated trial, a regulated lab, a GMP. All those things are too much of a heavy lift for where the industry is now. So the typical investor would shy away from kind of what we're doing. They would love to help develop drugs, and we could do the phase 1 studies and do good research on very promising drugs. But a personalized cell therapy product, that is the way paradigm has to change about how that is invested, infrastructure built, the deliverable, who pays for it, is it affordable? Our angle is a little bit radical. We have an angle. It might be popular with your audience, which is we kind of feel that cell therapy or cells, if it's your cell,
It's
you should
customized. It's made
your
for
cell.
you.
It's yours. You should be able to afford it. You should be able to decide how it should be manufactured. Do you want a CAR T working on it 1 week, and then if the leftover go to a peripheral TIL, or try different fractions all at the same time? Do you want to work with different companies to bring a construct and licensed for you based on your tumor markers? And so that's kind of some ideas we're trying to advocate for, to kind of experiment with some business models around how could this be funded. But at the end of the day, I think cell therapy, autologous cell therapy with individualized patient's own cells, they should own custody. They should have choice. They should have the right to try. Two parents who are trying to conceive a child and need in vitro fertilization, that's their embryo. They're paying for it. Even if an insurance company denies it, the patients can save up their money and they can choose to do that. So what we're saying to your audience, Chris, and you would agree, is your audience of stage 4 patients should be able to choose that as an option to right to try, as long as it's in a regulated FDA approved trial with a regulated FDA qualified clean room with a regulated credential,
Appropriate oversight, and
doctors, and everything's auditable FDA. Patients should have a right to own custody of their cells, engineer their cells so that they can have a chance to see if their immune system can be reinvigorated and chosen the right fraction to fight their own cancer. And so that's sort of where we hope the industry will be in 5 to 10 years.
100%. I mean, I say this across the board, no matter what disease you have, cancer or whatever it is, but I even compare it to something as simple as having like the flu or the cold. But if you get sick and, you need to take a medication to help you get better, and would you rather be able or have the choice to do one treatment one time or multiple treatments over a period of time? But I see one of the biggest problems is a lot of people just don't even know that those one-time treatments even exist, period. And that's the whole reason why I do what I do, because I was actually qualified to do what I did 9 months before I did it. I just wasn't told about it, wasn't knowledgeable about it, and I learned by default. But at the end of the day, I'm paying my doctors. I would figure they would work for me in a sense and be open to telling me. But at the same time, I do understand that not everybody can know everything that's everywhere all the time, but you gotta self-advocate, but if you at least know to ask, like, what are the clinical trials that are going on? What's out there, or how do I find it? But that all depends on how much you know at the time of diagnosis, and then also where you're going on what they're able to tell you. But just on my personal experience, if I was asked at the very beginning when it all started for me, hey, like, it's not the greatest odds, nothing has the greatest odds, but would you like to try to do one thing one time and it might work? And then if it doesn't, you can try all these other things. But that right there kind of leads me into what I want to talk about a little bit more is, and you mentioned this earlier, is T cell exhaustion.
Mm-hmm.
The more treatments you do before, like, as you go down the line, the more beat up your body gets. It's harder to fight when you've gone, like, 15 rounds than as compared to like the first couple.
Yeah, that's a good leeway into two good points that you remind me to talk about. The first is you mentioned awareness for the typical cancer patient, especially one who's metastatic and feels like they're overwhelmed.
Scary.
They don't know their options. Most oncologists are good intentioned, of course. They work in a hospital system. A lot of that is driven by the NCCN guidelines, which is all appropriate as what's proven best by the cookbook recipe of what we've done for many decades, building upon improving the standard of care based on the latest phase 3 trial and the latest commercial drugs that were approved by the FDA for commercial use, that's the standard. 99% of most agree. So the problem is there's always going to be pretty much all metastatic solid tumor cancer patients will eventually run out of the standard commercial drug options, hopefully not too fast, but many of them do. And if they're not starting to learn where else can they go for access to clinical trials or access to some of these cutting edge therapies on research trials, they may just be stuck in the same system, getting the same chemotherapy, getting the same different versions of that same class of chemotherapy for many years. And if they wait too long, to your point, their immune system gets so low that exhaustion is kind of one of the words. Exhaustion is when the tumors get exhausted or the immune cells get exhausted from fighting. In this case, if too much chemotherapy, the immune cells just get depleted. They're just donezo.
There's
They're
nothing
not
there
even
to work
exhausted.
with.
They don't even have a chance to get exhausted because they never fought anything. They just get so much chemo piled on. That's the mechanism of chemotherapy is it kills anything growing fast. Tumors might be growing fast. It works. It gets you out of a pain crisis. It's great. Your immune cells are growing fast. It works. It taste buds and, bone marrow grows fast. That's why you lose your hair, et cetera, et cetera. The problem is though, and this is the concern I see amongst a lot of the patients who come to our clinic looking for phase 1 studies, some of them have had 2 years straight of just chemotherapy back to back, 2.5, 3 years, 5 different regimens. And it was almost too late when they came looking for immunotherapy studies because they had no immune system left. It's very hard to reinvigorate or find any TIL or much less peripheral TIL or reinvigorate or a vaccine to work when you're immune depleted because you've had 2 straight years of chemo. It's hard for modern immunotherapy to work. So I think that the 2 points, awareness, patients should, as soon as you're stage 4, yes, if you're in pain crisis, go get your few rounds of chemo. Each time you get a dose of chemo, it's a logarithmic reduction, generally, if it's sensitive. You'll get out of a pain crisis, get back to your good function. But as soon as you got the tumor under control initially, start looking for where are the cutting edge immunotherapy trials in your region, personalized vaccines, oligosense nucleotides, all those kinds of things. And the place to find that is on clinicaltrials.gov. And most of your viewers hopefully know this by now. If they don't, this is a good teaching. It's a federal law that clinical trials are listed there by all sponsors, all phases, all disease types, not just cancer. So, and you can go on there and you can select, I have stage 4 melanoma. I want to know what trials are in my region, by radius, by map, or how far you want to travel, who's offering them, what the sponsor is, even the phone number of who to call to get on that wait list. And those motivated patients that do that, they often are on 3 and 4 different wait lists at 3 or 4 different facilities in their tri-state area. So they're on the wait list for when slots open for these studies. And that is an easy, low-hanging fruit to just go to clinicaltrials.gov, type in your tumor type, your stage, and look and see what's available to you. And then you want to do that so you're on the radar for these trials to get access before you have 3 straight years of chemotherapy, and don't realize, oh, maybe I need an immune system to have a residual immune function to be able to get a good benefit from an immunotherapy trial. So if that's a take-home message, that's well worth it being stated here.
What about the CRI search engine, Cancer Research Institute? I had a metastatic breast cancer patient that lives in San Antonio that wanted to go to Maryland to do TIL at the NIH, but she wasn't qualified because she had an autoimmune disease. But she's a 10 plus year metastatic breast cancer survivor, continuing to try treatments, but she's got a little bit of a clinical background. And one of the things that we were talking about was the multiple search engines. And there's definitely clinicaltrials.gov, but she
Yeah,
also highlighted and brought up the Cancer Research Institute, the
yeah, yeah.
CRI, which has been
Great.
around since the 50s or 60s, I think she mentioned. I mean,
Yeah.
don't quote me on that, but
No, no, that
it's been
that's
around quite a long time.
they also on that site, they don't list everything like clinical trial. I think they favor cell therapy studies and more of the cutting edge. Yeah, that's good. I've looked at their program. Most of our phase 1 studies are listed on both entities. So yeah, those are really good points. I think I've been doing phase 1 research for 20 years, and the typical patient now that is seeking out trials, they really are taking their care into their own hands more than you've ever
You
seen.
have to.
Googling, they're learning, they're doing all these things. They're bringing a 3-ring binder of all the different studies they want access to. They're going to the websites of the biotech companies to read about the different pipeline, where they are, and they're learning. They're doing their due diligence. And I think that probably makes them feel like if they can take some control of
Em-
what they
Empowerment.
can seek, yeah, it empowers them. It gives them some hope, and we love meeting patients like that because when they're motivated and have a good support system and understand what immunotherapy is about, and I think that's a good match for the typical type of patient. We have so many phase 1s that we say we're a rapid fire clinic. If the first study doesn't work after a couple months, we'll rotate into a different study and a different study. So we try not to run out of options as long as patients are strong enough and motivated and willing to come and have good organ function. We will continue to offer them different trials and work very closely with their primary med onc.
Yeah. No,
We're
I
on
mean,
speed dial with like over 100 med oncs here on the East Coast.
that's huge because for the average, just anybody out there that has never had a diagnosis before, had to be a caretaker, or caregiver, or known somebody specifically that has gone through any sort of cancer or rare disease diagnosis, they only know what they know. And
Yeah.
so I always tell people this, and it's, now I'm coming up on my like 7-year anniversary of my diagnosis, but the only thing that I assumed or thought of when it came to cancer, period, was cut it out and you do like chemo and maybe radiation, but that was literally kind of it. And once I was recommended and got to a specialist and they kind of confirmed it, they told me that chemotherapy for many, many years was always like standard of care. But for a metastatic stage 4 patient, it's a band-aid. If I was to ever to need to use it, it would be a temporary thing. It was never an end game solution. And I had to use chemo a couple of times to kind of pause things as they were getting out of control. But the whole concept of immunotherapy, of, being specific and being targeted, kind of planted that seed in my head of like, okay, wait a minute. So you're just not trying to kill everything. You're trying to actually go for what's on the target so I can aim at the bullseye. It's constantly evolving, it's constantly changing. When I was diagnosed, and it had been, I think, 1 year or so since CAR T had got approved. It had been maybe like 6 or 7 years since IPI was approved, or aka Keytruda. And since then, there's been multiple additions and combinations, and even the advanced cell therapy clinical trials in May of last year. This is when I met Tom Whitehead in person. We were at this Forensic Cancer Research and Parker Institute 1-day cell therapy meeting. And the whole point of it was, at the time, there was 7 approved advanced cell therapies. 6 of them were for hematologic cancers, and 1 of them was for solid tumors. But 10% of all cancer is hematologic, and 90% of it is solid tumor. And even though all these treatments have been in progress in clinical trials for decades and decades, getting them through the whole FDA process to finally get them approved and make them available, it's like uncharted territory. And that's, what that meeting was about. We continued again this year in these conversations are constantly happening. And it happened last week, but I finally watched it 2 days ago. But did you catch the FDA roundtable? I caught it on a recording of it. I didn't get to watch it live, but it was a 3-hour recording with the Health and Human Services. There was a lot of Carl June, Crystal McCall,
Yeah.
a lot of key players in the cell and gene therapy field were there. And I've heard the FDA say this to me in person, but they're willing to talk. They want to, help.
Yeah.
They know the changes
Yeah.
are here and want to help make those possible. I mean, did you get to see
I
any of
saw
that?
the high level review summary of it, but I think all that's good momentum. I just saying something, but then not seeing it happen. I mean, I'll be a
It's
believer,
frustrating.
but it is the incentive now is they're going to have to keep up with Asia and Europe, who they're already pivoting towards
And they mentioned that.
decentralization, cell therapy. They're kind of defining what is minimally manipulated, and autologous cells that are not gene modified are probably safer compared to like a gene modified cell that's maximally manipulated. So hopefully the regulations will loosen up a little bit for autologous cell therapy that's like TIL or peripheral TIL. So I hope so. The other thing that you said reminds me to bring up another point for your listeners, which is kind of a good hope point about immunotherapy. If you say the word surgery and immunotherapy in the same sentence, this historical standard used to be when you get cancer, surgery, dissect all the lymph nodes, and then along came, well, let's give chemotherapy first, shrink the surgery. So when we cut on the surgery, we're not cutting through a growing living tumor. And then that data seemed to look a little bit better. So that became a shifting standard. Neoadjuvant chemotherapy, then surgically resect it. Where the thought leaders and where the data now is shifting, and this is incredibly promising. Turns out, if you give immunotherapy while the tumor is still in your body and then go for a resection, sometimes the tumor is gone by the time you resect it.
Yeah,
It's so like basically simple. The TIL that were in the tumor just needed a little bit of immunotherapy to work better. And the lymph nodes that were around the tumor do better if you don't take them out. I mean, this is like,
I cannot agree with you more on that.
that was our
100,000%.
That was our dogma for like the textbook, cookbook, guidelines for a long time. In almost every study you see now coming out where they do neoadjuvant or immunotherapy before, it's way better. And we're seeing survival and durability is better. So it's telling you something that the TIL in the tumor and the lymph nodes that you don't take out matter if you just time it right. And we're kind of hoping the peripheral TIL are there. And I hope in the future, in 5 or 10 years, we'll see something where you can just do peripheral TIL and immunotherapy and not have to resect as many cancers. The trials have to be done. We got to, make sure we stay that's not the norm, but that's where
No, absolutely.
hopefully in the future, there'll be a cell therapy facility like ours across the street from every major hospital. All TILs banked. If you have to have surgery, at least bank your TIL. Don't just throw the tumor in the bucket, and nobody banks their TIL unless they're going on a study. So that all needs to change, and we should be banking peripheral TIL as well and looking at more trials for that. So yeah, so that's a good reminder.
I wanted to ask you, because I remember you telling me this at one point, but say, for example, someone, is enrolled and they're signed up and they're doing peripheral TIL. How often would they need to come back for an additional injection or to be seen? Kind of give me just like a 101 layout of how
Right now,
that goes.
like these autologous cell therapies like NK cells, peripheral TIL, gamma delta, all these autologous peripheral derived polyclonal immune cells, generally they are kind of repeat dosing. For our model, just for how we envision it, we're envisioning the hopes. We haven't dosed anybody yet. We're submitting our IND here in the next hopefully 6 months or so. We're anticipating that patients may leukapheresis once, and then we may dose them every 2 weeks. And then after 2 months, if they need, depending on the scans and what we're seeing response or not, we may leukapheresis them again to create a different batch or a better batch or a different fraction.
Be like an updated version of what their cells currently are at.
Bingo. Or leukapheresis first to get a big batch to kind of give a big dose, and then do whole blood to do maintenance or iterative dosing on medium scale. So large scale leukapheresis, medium scale whole blood, kind of like to iterate or steer the immune response and measure it as it goes. And so we're now working on different preclinical models for that and actually writing the protocol as we
That's
speak.
cool. It's mega
So
cool.
so stay tuned. I think in the next, 5 years, people will be hearing a lot more about peripheral TIL or peripheral or reactive cells or exhausted reinvigorated cells, and how potent the immune system is if you can capture it and you can expand it and if what it targets, and then you've got the target, in your petri dish, and you can pick which clones do the best killing. Yeah, and you might not need to do the surgical resection if you've got these trial options.
So that's what they call in vivo, like instead of ex vivo. Is that
Maybe a little.
correct?
So it
Do I have the
depends. So,
terminology right?
depends on how you define it. So right now, most of the cell therapy is we take the cells out ex vivo outside the body. We grow the cells, give them back. Like you got your own cells back. So that would be ex vivo back into your body. There is another paradigm shift going on now, not with polyclonal cells or cell therapy, but it's with the gene construction. We talked about the gene constructs and putting in the gas pedal in the specific like CD19 CAR T. There is now a paradigm shift where maybe about a half dozen companies are working on don't take the immune cells out, instead just put in the gene construct directly into the patient. Let the gene construct have like an envelope and find the immune cells, and then the immune cells get the gene construct, gets in them, and turns on the gene on the surface and the gas pedal to go find and target to fight the cancer. So that's in vivo. So it's like cell therapy, but the gene edits or the gene constructs
They're
are
done within the body without having to take it
correct.
out of the body.
So technically, you don't need a cell therapy factory like I got behind me. You just need to know how can you engineer a virus or a lipid nanoparticle to carry the gene and get the gene into the cell. That's a whole other discipline of
No, yeah, no.
but that's where those trials will probably start to get to be reported out in the next year or two. So if your listeners want to learn about in vivo, um, you know, cell therapies or in vivo CAR T, gene editing. Just Google those things, and you start seeing some of those early phase studies get reported out in the next year or two.
There's a couple of dudes that I met at excuse me one second, at Advanced Therapies Week
Yeah,
for Facilitate here in Dallas in January, and it was Ben and Jeff, and they do a LinkedIn live like every Thursday. I think this week it's gonna actually be tomorrow instead, but I came on and did, I think, like their second episode. They were talking about TIL, but I follow those guys, and they specifically in one of their episodes recently, and I have to ask them to chime in on this, but we're talking about the NK cells and doing the in vivo versus the ex vivo. But for someone like myself, I just look at it in simple terms as that's one less step for a patient. You know what I
yeah,
mean?
yeah,
And if it can be
yeah,
done like that, like that, just to me, it just makes more sense. It's just
yeah,
like if I were to have to explain it to my youngest niece or nephew on what's going on or how it works, like I want to be able to tell them that. But I always try to bring those kind of topics up because logistically for a patient out there, when they're trying to think and decide on what kind of treatments are out there, what to
yeah,
do, their notion of clinical trials and everything. I admit openly, I had it all backwards 7 years ago. You know what I mean? I thought I had to do everything that was approved first before I tried a clinical trial. And knowing what I know now, like I said, I had it backwards. I would definitely have tried
yeah,
something in the trial space. I know numerous patients, survivors that I've met that are now survivors because of phase 1 trials. So I can't stress how important and how critical that is, because those efficacies might be the same, if not a little bit more sometimes, but you don't know unless you try. It still comes
yeah.
back to that whole point.
Yeah, no, a lot of research trials don't even meet their accrual goals for many reasons. So it's awareness, it's patients willing to travel, being motivated, and it's a little bit scary volunteering for a
It's
trial.
always scary, man. Like, that's why I try to talk about it, you know what I'm saying? And kind of
Yeah,
humanize it a little bit more and just be like, I'm not the smartest cat on the block, trust me. But I was willing to take the risk, and everything's risky.
I just feel like everybody that has stage 4 cancer that's incurable deserves access to the cutting edge. And
100%.
if they just get 1980s chemotherapy as the only thing offered to them, I
And that's
feel
all.
they're not aware and they're not being, I get it. Their circumstances may vary for different people, but I just think they should at least be aware that hopefully in their community, in their region, they can get help, transportation to go learn about what else is out there. Because the trials do exist. It's just a
You
leap
got to
of
know how to find them.
and be willing to drive sometimes and talk to your
Yeah.
oncologist or get another opinion. But yeah, all good points, Chris. And, I love to see you doing well. And I look forward to more of these, episodes. And at one of my future talks, I'll have to show you some of the studies on the vitiligo that we worked on years ago, how favorable that is and how lucky you are that you got so much
I
vitiligo.
mean, yeah, I think I might have mentioned this to you, but when I was first started getting the vitiligo, it was kind of weird. It was like real patchy, and it was just
Yeah,
kind of like around this goatee area. And this was
yeah,
before I did TIL. This was just doing combination immunotherapy and such. But one of the things that stuck out was my eyelashes, because those were one of the first things to change color to white. But after I did TIL and I completely lost like all my body hair except for maybe like a little bit of my eyebrows and it grew back, it was bright white. And I was a little
yeah, yeah,
self-conscious about it initially. So when I went to go get like my first haircut in the summer of 2020, I had my hair dyed because
my gosh,
I just mental health wise, I just felt weird about it. And
yeah,
then I was able to kind of get through it. And then I was like, okay, well, I'm not going to dye it again. But I have some pictures from that era to where I have this two-tone hair where it's brown on the top and white on the bottom, which is like opposite of like what I looked like in the 90s, where I
yeah,
had brown on the bottom and like blonde tips on the top, because that's
yeah,
what you
yeah,
did
yeah,
and stuff. But it kind of became a part of me, became my identity, and I learned to embrace it and then love
yeah,
it. And when it finally grew out and cut it, the white eyelashes and just my white hair color, period, in inclusion to my age, I just sparked up so many conversations that I would have probably never have had at the most random places with the most random people, to
yeah,
where then I could like tell my story and then talk about my treatment. And then before you know it, like it, that's kind of how it spread. But I became extremely proud of the loss of my pigment and my white
yeah,
hair and my
yeah,
white eyelashes.
yeah,
And it's confusing sometimes to a lot of people that I haven't seen in a very long time, because they remember me as one way, but then I start talking and they're like, oh,
yeah.
okay, I remember you now.
That's
But I always try to remind people, I mean, that's a good sign when you have those changes. You got to look at the positive of it. And man, I couldn't imagine myself ever looking the
yeah.
way that I used to. It's like a whole new Chris, you know what I mean?
Well, I'll look forward to showing you some good animal models of the research. Just anybody in the tumor immunology research space, melanoma taught us so much, and breaking self-tolerance to not only kill the melanoma, but the pigment cells as well. Those are the durable responders and how that happens. So yeah, I'll show you plenty of many different animals in the animal kingdom can break self-tolerance with vitiligo, cure themselves of cancer. It's just remarkable. It's a great story, and it'll be more inspiring to tell you. So you can realize it's worth it. It's so worth it. You get some
So
vitiligo. It's on TV. They're advertising how to get rid of vitiligo, but people with melanoma, it's their holy grail, to get it. So
So this is the first of our conversations. We're going to have about 4 more or so within the next year. But what I want to ask you now, just based upon this conversation today, is off the top of your head, what is something that we should focus on the next time we get on and do this that
think
we should
that
highlight?
we talked a good amount at the broad level. I think your audience might like to know kind of what's going on in the industry with hurdles. And these are things that kind of come up at Deloitte, like what can the industry do better? What can we as a society prioritize? And then it comes back to maybe motivated cancer patients can actually change regulations. The HIV community changed regulations
Exactly.
Women with breast cancer can change regulations. Metastatic cancer patients should be able to demand some regulatory changes so maybe autologous cell therapy can be easier to get access to, more affordable and decentralized. And I think that those would be some good topics. And then maybe on another talk, we'll do a deeper dive into the biology. And I'm happy to show a couple slides and about
Yeah,
how
dude.
immune cells work, the different fractions, so people can get a little, 101 on cell types of the immune system. That'll be good. Yeah. We'll come
No.
up with some good stuff.
100%. Like, I got to admit, I kind of held off for the longest time, but I recently acquired the paid for version of like Chat GPT. And I'm
Yeah,
like, oh, I love it because it was annoying as hell for me to be able just to ask once a day and then have to wait another 24 hours. But I'm like, it was building upon what I was doing. And this is a whole another thing, but one of the influences was from BioBridge Global Advanced Therapies. They put out a bi-weekly blog and they have graphics that go with it. And a lot of people like myself, I mean, I'm a visual learner. So you take somebody that knows nothing about cell therapies, advanced cell therapies, cell and gene therapies, cancer, rare, whatever. But when you show them graphics and
yep,
then are able to explain, I'm like, oh, that just makes so much sense. So I'm learning to try to embrace that and then incorporate that
it's good. Yeah,
into trying to share it and make it
yeah,
more normalized. So, but like this super cool, man. Like I love what you're doing. I'm loving the fact that we, met and connected, and it's like I think it again, my eyelashes, you know what I'm saying,
yeah,
is
yeah, yeah,
probably one of those things where it was like we end up sitting down, shooting the shit and chatting, and here we
yeah.
are.
Yeah. No, this is a great story to tell. Your good luck with your continued podcast and web channel. And I'm happy to be a champion for what you're trying to get the awareness out and piggyback on raising awareness for what we're doing. And happy to do some more teaching. And yeah, I keep inspiring everybody. And I got to go check with my nurses now before they close the clinic and make sure
No, yeah,
everybody's
I
yeah.
got you. Well, for everybody listening, thank you very much, and we'll check in with you soon. Peace. All right, that concludes today's episode of TIL Talks with Chris White as we talk TILs, tumor infiltrating lymphocytes, and all advanced cell and gene therapies. As a reminder, I am not a medical professional or medical expert. I am only a professional and expert on my story and my experience. If you are needing or seeking medical advice or assistance, please reach out to your care team. I'd like to thank my guest today for joining me on TIL Talks. To our listeners who have not yet done so already, please follow, comment, subscribe, share, and smash that like button. For more information about TIL Talks and related resources, please visit www.tiltalks.com. I'm Chris White. As always, sending love to everybody. Peace.